Hello, my name is Cyril Jacob Joseph, and the date is November 15th, 2023, and this is my presentation on mitosis. Thank you. Mitosis is part of the cell cycle, and in eukaryotic cells, the cycle is divided into two distinctions, interphase and mitosis. Interphase is characterized by the G1 phase, otherwise known as the first gap. The S phase, synthesis, and G2 phase, second gap. By following the nuclear DNA during interphase and mitosis, we get a clear understanding of this process. In G1, the DNA is in the nucleus, and it is relatively relaxed, and meanwhile the cell is metabolically active. In the S phase, here we have DNA replication, and each pair of homologous chromosomes will duplicate into having two sister chromatids, with cohesion attaching the sister chromatids together. In the G2 phase, the nuclear DNA is starting to condense. However, they are still not visible using a microscope. Next, we have mitosis. Mitosis consists of 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. In prophase, the nuclear membrane begins to disintegrate and disappear and remains gone until telophase. Meanwhile, the sister chromatids are beginning to condense, and they are now visible using a microscope. In this stage, we can see that the centrosome will begin to migrate to opposite poles of the cell, and a collection of microtubules will form between the two pairs of centrosomes, forming a mitotic spindle and an aster. In prometaphase, the chromosomes are now fully condensed and are making their way to the metaphase plate. Microtubules from the centrioles, called kinetochore microtubules, will attach the kinetochore on the sister chromatids. In metaphase, the sister chromatids are now aligned at the metaphase plate. It is at this stage that all the sister chromatids are attached to a spindle fiber. Moving on to anaphase. The sister chromatids separate and move towards opposite poles of the cell in a process called disjunction. This is achieved with the help of motor proteins which utilize ATP in the process. As a spindle fiber retracts the part of the sister chromatid to its respective poles, the cell begins to lengthen. And now with telophase, the nuclear membrane reforms each set of chromatids, which can now be classified as daughter chromosomes. The chromatids uncoil back into chromatin, and the spindle fibers begin to deconstruct. Now, on to the differences between mitosis and cytokinesis. Mitosis is karyokinesis, in that this concerns the division of the nucleus and its content, whereas cytokinesis is the division of the cytoplasm, which leads to the physical separation and splitting of the cell into two separate daughter cells. This manifests differently in animal and plant cells. As animal cells form a cell furrow using a contractile ring made up of actin and myosin filaments, plant cells form a middle lamella, which is a ridge that forms along the line of the metaphase plate, which develops into a cell wall separating the two new daughter cells. Thank you.